Hi, I'm Frank Rattacco from Thermo Fisher Scientific, and I'm happy to talk to you today about some new approaches and technologies that we are using to enable accelerated development and manufacturing timelines for biopharmaceuticals. Thermo Fisher Scientific is a leading global biologics CDMO, supporting our customers with drug substance and drug product manufacturing facilities at locations around the world. As a company, we have over 30 years of GMP manufacturing experience. We focus on mammalian expression systems. Our capabilities include cell line and process development for early and late-stage molecules, and clinical and commercial manufacturing with over 32,000 liters of single-use GMP capacity currently. As you can see from this slide, we have extensive experience with tech transfers and development programs for multiple molecule types, and we can offer integrated gene-to-patient solutions, including drug substance, drug product, and Fisher Clinical Services to provide all of your manufacturing and logistical needs under one roof. The past few years have seen a dramatic growth in the number of approved biologic therapies, and each year, more and more biologics are given a fast-track designation or breakthrough therapy status. As a result, biopharmaceutical companies are looking for approaches and solutions to enable accelerated development timelines and a faster path to the clinic for their breakthrough molecules. There are a number of critical items to consider when trying to reduce development and clinical manufacturing timelines. The goal is to balance speed, risk, and robustness, understanding where you can take appropriate risks in development, while still generating the quantity of material needed to enable your clinical trial and without sacrificing product quality. In order to address these needs, Thermo Fisher recently launched the Patheon Quick-to-Clinic offering. With Quick-to-Clinic, we can save 7 months compared to a traditional program, providing released GMP drug substance in just under 14 months, or released drug product in 15 months. Quick-to-Clinic leverages our deep experience in biologics, combined with our in-house platform processes and raw materials to provide our fastest timelines to GMP drug substance or drug product release, specifically for IgG1 and IgG4 monoclonal antibody molecules. These development timelines are enabled by the use of well-established platform processes for monoclonal antibodies, high-throughput automation technologies such as the Beacon, Amber, and TCAN systems in our development labs, multiple development activities conducted in parallel, and standardization of raw materials and documentation to streamline development and manufacturing while minimizing risk to product quality and regulatory filing. The Thermo Fisher platform for biologics development and manufacturing is flexible. We follow a very familiar process commonly used for monoclonal antibodies, similar to those used widely across the industry. We leverage Thermo Fisher cell lines, raw materials, and equipment wherever possible to streamline procurement and development activities, but we can also accommodate materials and equipment sourced from other vendors within our platform. For example, we can use Thermo Fisher single-use bioreactors, but we also have other manufacturers' bioreactors, such as GE Accelerex, within our platform. A quick-to-clinic program will deliver a cell line, a process, tox material, all necessary reports, and approximately 1 kilogram of released GMP drug substance for your clinical trial in 14 months, from DNA to drug substance. 15 months includes drug product manufacturing. This slide illustrates at a high level the quick-to-clinic timeline, with cell line development taking about 4 months, delivery of non-GMP tox drug substance at month 8, GMP drug substance at just under 14 months, and drug product at month 15. This slide shows a bit more detail of the overlapping activities taking place during the process development and manufacturing timeline. Cell line generation is performed using a Thermo Fisher cell line, shown here as FreedomCho S. Automated systems such as the Beacon, Amber 15, and Amber 250, and the TCAN Freedom Evo are used to streamline process development. After confirmation of the process at the bench scale, a 200 or 250 liter pilot run is conducted to generate material for toxicology and viral clearance studies, and to provide confidence in scale-up of the process to 500 or 2,000 liters for GMP manufacturing. Our platform has, until now, utilized the FreedomCho S cell line. But later this year, we will be launching Freedom ExpICHO S, a new, higher titer, stable cell line system for expression of biologic molecules in CHO cells. This cell line is based on the very popular ExpICHO transient expression system, but has been adapted for stable expression and high titers with an all-new vector system. The system is designed to provide antibody titers in the 3 to 5 gram per liter range within a CLD timeline of 4 to 5 months and without royalty or milestone payments, just like the existing Freedom CHO kit. Here's some data showing how Freedom ExpICHO performs. The bar graph on the left shows a comparison of monoclonal antibody titers in ExpICHO-S compared to an HEK 293 cell line and an older CHO-S cell line. The differences are quite dramatic. The line graph shows titer data from 3 clones taken from a single ExpICHO-S transfection, with monoclonal antibody titers ranging from 2.5 to 3 grams per liter in this instance. Further, we have observed consistent product quality between transient and stable expression of a molecule in the ExpICHO system, allowing our customers to confidently move from transient expression at the R&D stage to stable expression for development of a manufacturing process with low risk of significant changes in product quality. And lastly, I want to highlight that the Freedom ExpICHO kit will feature a new 2-vector system, which will allow for good control and tuning of heavy and light chain ratios to optimize antibody expression. Here's some scale-up data of a Freedom ExpICHO-S cell line using Gibco's stable production medium optimized for it, called ExpICHO SPM, showing good growth and antibody production in a Thermo Fisher single-use bioreactor without any process optimization or loss in titer compared to the bench scale. And for our cell line development programs, including Quick to Clinic, we are now utilizing the Berkeley Lights Beacon platform for cell line development. This is a very exciting technology, which we were one of the first CDMOs to offer to customers. The Beacon performs single-cell cloning on a chip, using light to move single individual cells into pens holding only 1 nanoliter of media. And the system can visualize growth and antibody production at the nanoliter scale. This instrument gives us the ability to evaluate 7,000 individual clones at once with assurance of monoclonality, allowing for faster cell line development timelines and the screening power needed to identify the highest producers in a population of transfected cells. I hope you've enjoyed this presentation. Please feel free to reach out to the experts at Thermo Fisher Scientific if you would like to learn more about our capabilities as a leading global biologics CDMO. Thanks for your time.